good afternoon. So it's my pleasure indeed to introduce the speakers and also very briefly in the next few minutes the subject they will be talking about, which is the role of mechanical forces in tissue organization. So here in the slides you might recognize pictures from a classical book by Darcy Thompson on growth and form that's already a century old now, and which was one of the first one to notice the striking similarity between tissue organization and some passive physical systems such as foams here. And this is a beautiful book. This was really influential, but of course, it's still largely qualitative, right? You see that there's a lot of hexagons in there and a lot of hexagons in there. But it took quite a bit of time to actually to actually put a bit more of a quantitative foundation behind all of this. And indeed, even though I think just from the laws of physics, we know that any deformation that happen in embryogenesis must have underlying physical forces. The real question is which ones, right? There's lots of forces that can be generated in lots of places. And answering that was really the start. This really started to be quantitatively addressed in the 80s by classical studies, in particular by the group of Oster, Keller, Albrecht, Murray, and many others, which had in common to essentially try to really look in detail at some morphogenetical process and try to infer the nature of forces from essentially deformation fields. So this is a paper I really like, for instance, in the 90s on how sea urchins invaginate. So you can see here in this microscopy images that this part here is becoming a bit thicker while morphogenesis is proceeding. And what the authors do here is not just to do sort of qualitative observations, but they really try to compare lots of different models. It's surprisingly prescient work where they try to understand. They try to compare 5 different models with 5 different nature and localization of forces. And even though all of them give rise to qualitatively an invagination, you can see that if you look in detail at the deformation fields of cells, there actually are a number of subtle differences. So here, and you can really use this to try to explore different models. For instance, could it be that cytoskeletal tensile forces in different places drive these deformations? Could it be instead compressive forces from the swelling of an ECM which is below? Could it be my favorite name, which is a cell tractor model, where you would have cells moving towards the region of the invagination and driving compression and buckling? These are the type of questions that people started asking in the 80s with these studies. This was also, of course, triggered by advances in biology, in particular. A bit before actomyosin had emerged as a prototypical example of cytoskeletal force generation in cells. Indeed, it was discovered in muscles in the 40s, but then its generality in force generation of non-muscle cells was found in the mid-70s. And so there has been a number of classical studies, in particular in Drosophila, that implicate subcellularly generated actomyosin forces that are transmitted then by junctions throughout the tissue, and then sort of drive sequential events of bending and deformations in epithelial sheets to end up sculpting the embryo. And in this kind of picture, you can see sort of the embryo patterning as providing some type of blueprints for forces that are then the effectors of making this type of origami. I really like this picture from a recent 2016 review. So this is one example, for instance, of forces. One example of forces from the cytoskeleton that can drive embryogenesis. There's been, of course, a number of other types of forces that have been reported, and interestingly, many of them already conjectured again by these papers in the 80s and 90s. For instance, the idea of active migration forces acting through in the context of condensation of the mesenchyme, for instance, growth forces that can give rise to buckling instabilities, forces from the ECM, forces from ECM swelling that actually sort of deform the monolayer, or changes through the passive resisting forces of embryos to help sort of to sculpt it. But interestingly, and sort of particularly relevant to us, mechanical forces don't. It's become apparent that mechanical forces don't just shape tissues; they're not just effectors of tissue deformations, but they can also transmit information exactly in the same way as morphogens. Actually, you can make. There's actually a very strong and formal analogies between the information that's propagated through diffusion of morphogens from a source and the diffusion of mechanical stress or strain from a point from a point force. And because of that, in many of these cases, it's now being realized that mechanical forces indeed are not just effectors that would simply follow a developmental blueprint, but they can really give rise to complex feedbacks at a cellular, subcellular, cellular, and supracellular scale, which can give rise to self-organization, which is one of the big questions, I think, in the field that we'll hear a lot about today, which is actually an old question, for instance, which was posited here by Albrecht in a beautiful paper on the logic of monsters, but which is really which we can start really to address nowadays with contemporary tools, I think. And in particular, one force which I haven't mentioned now, but which both of the speakers are going to specifically talk about, is something that has emerged, particularly in the last few years, is the idea that the lumen, that the extracellular fluid itself, not just cells, can play key developmental roles, both by providing diffusible factors such as morphogens, but also providing forces themselves, both in the shape of hydrostatic and osmotic pressures. The forces from the fluid have both drawbacks and advantages. One of the drawbacks, for instance, is that fluid pressure is necessarily isotropic, so it's exerted in sort of all directions. So you can't have, for instance, directional shaping, such as for active migration or patterns of continuous extension. But it has quite a few advantages. For instance, osmotic forces can be really large. Because of fluid flows, you can have relatively homogeneous forces in large spatial domains, which can conceivably help, for instance, with coordination. And because it can also transport a lot of things like diffusible signals and morphogens. And therefore, in that sense, it could be an ideal platform to integrate cues from morphogens, sulfate, tissue mechanics, local geometry, etc. And so, both of the speakers today will talk about some aspects of extracellular fluid, in particular, and fluid forces as a design principle of tissue organization. And I think both of their background reflect the interdisciplinary nature of the field. So Priska was trained as a chemist and a molecular biologist before getting more and more interested into the principles of multicellular self-organization, in particular in organoids, since she started her lab in 2015 in the FMI in Basel, where she recently became senior group leader. And Jean-Léon Maître has been interested in the role of forces in developmental biology since his PhD, first in zebrafish, and now in his own lab in mouse embryos and humans at the Institut Curie and in CNRS. And so I'm very excited to hand the speaking problem to them. Jean-Léon will start, and then Priska. I should say about questions. So they will talk. They will talk in a block because of the similarity, because of some synergies between their talks. But you can type your questions. There's a Q&A box at the bottom right. You can type all of your questions. You can also upvote for questions of others. And so after the two talks, we will sequentially sort of read the questions according to their most upvoted, and the speakers will answer, and then we will have a more informal meet the speaker session afterwards. All right, that's all I had to say. So Jean-Léon, I give you the stage now.
Thank you, Edouard. Okay, now you can see the slides, right?
Yes.
Yes, perfect. Okay, excellent. So thank you very much, Edouard, and thank you everyone for showing up. I think it's a very exciting format, and as you will see, I think there's a common theme which will come out of this series of two talks and discussion, and I'm looking very much forward to it. So as Edouard was mentioning, so we are starting to discover like the strategies that cells and tissues can adopt to shape themselves, and this is very much what we try to do in the lab. We try to uncover what are the strategies that cells have adopted to shape the early mammalian embryo. And so for this, we use the pre-implantation embryo as a system, as an experimental system, because it offers a unique opportunity to investigate the shaping of the mammalian embryo using live embryos. And so here, this is, for example, a mouse embryo, which is kept in vitro under the microscope. And you can see the zygote, which divides into a two-cell stage. So the the divisions are cleavage divisions where you have division without cell growth. And then the embryo will start morphogenesis here with compaction and will create this structure, which you can see here, which is called a blastocyst. And so the blastocyst is the structure which is going to implant the embryo into the maternal uterus. It has a very specific architecture, which you can see here, which is conserved between several mammals. And so, for example, here the architecture of the blastocyst of the mouse is very similar to the one of the human. This specific architecture is the following. So you have a surface epithelium, which is called a trophectoderm. And this forms an envelope, which encloses a fluid-filled lumen called a blastocell, which we are going to talk about today. And then a cluster of pluripotent stem cells, which are here, which is called the inner cell mass. And the inner cell mass is going to give rise to all of the cells of the mammalian body. The trophectoderm will only contribute to extra amniotic tissues of the placenta. The job of the trophectoderm is to inflate this lumen, attach to the uterus, and then contribute to tissues of the placenta. And so, as I mentioned, we are interested in general in the shaping of this structure. And today I would like to discuss the formation of the first mammalian lumen, the blastocell. So this is here an embryo before the formation of the blastocell. It is fairly symmetric. There are inner cells and outer cells. And at the end of the formation of the lumen, what you can see is that the embryo has broken its radial symmetry, and it now has an axis, which is formed by, on one side, the lumen, and on the other side, the inner cell mass. And this needs to be robustly made every time. And you may need to make sure that some of the contacts are broken, but some of the contacts are maintained. You do not want the lumen to form within the cluster of inner cell mass cells. You want to make sure that the inner cell mass cell stays cohesive and is not scattered throughout the blastocyst. Otherwise, you may get multiple embryos, which you may or may not want, but you may want to control. So how does the embryo robustly establish the lumen at the interface between trophectoderm and ICM cells? So a little bit about the cell biology of lumen formation. So the way that animal cells make a lumen is by having a polarized epithelium. And so that is the trophectoderm, which is at the surface of this embryo. It is polarized in the sense that it has an apical basal polarity. The apical here in red is at the surface of the embryo, and the basolateral side of the cell, which includes the cell-cell contact, here is here labeled in blue. And so this polarity is allowing polarized transport of osmolytes, which is mostly salt, into the intercellular space of the embryo. And those salts will then be retained within this intercellular space thanks to tight junctions. So specialized junctions, which make sure that the epithelium is tight, and they are here labeled in green. And so because of this, the salt will accumulate within the intercellular space, and this will attract water. The osmotic gradient will attract water within the intercellular space, and this is what will lead to the accumulation of fluid and the inflation of this lumen. So as I mentioned, what is not clear is how does the embryo ensure that the lumen robustly forms at the trophectoderm inner cell mass interface. And this will set the first axis of symmetry of the mammalian embryo, which is important for further development of the embryo. Okay, so to understand this a bit better, what we did was simply to just image the process at high temporal and spatial resolution. And so this is a mouse embryo at the onset of lumen formation. It is now 32 cell, and it will start the formation of a lumen. So what you can see is that there are small gaps which appear within the tissue throughout the embryo. So there are small pockets of fluid which appear at cell-cell contact, including contacts of the inner cell mass. So the contacts are transiently separated by those small water pockets, which we call microlumen. And eventually all of those microlumens disappear until only one remains and becomes the largest dominant one, and that is the blastocell, the lumen. So how does the embryo robustly establish this phenomenon, even though early on it breaks all of its contact? So to quantify this a bit better, what we did was to follow those microlumens, and we classify them in two different types. We have the ones which are strictly contained between the cell-cell contact of two cells, and then we have the ones which are at the corner of the cell-cell contact, which are here labeled in blue. They follow the same kind of dynamics with slightly different magnitudes, but basically they grow and then they shrink, and this takes about 5 to 6 hours. And then the only one which remains, which does not shrink, is the blastocell. So to look into this, we got very much inspired by a study from the lab of Chabit-Repat, where it's a very different setting. They had plated epithelial cells onto a hydrogel, and they flushed some medium through the epithelial layer. And what they could see is that this would fracture the epithelial layer. So this is hydraulic fracking. So the fluid is just crushing into the epithelial layer and just breaks apart the cell-cell contact. And then the cells are repairing those gaps in the tissue. And so we thought that those structures resemble very much what we observe in the embryo. And so we looked into the possibilities that hydraulic fracturing would be at the origin of the fracture of all contacts within the embryo. And so we measured the pressure of the fluid. We saw it was in the correct range of what is causing the fracturing in vitro. And then we looked into the adhesion molecules, which must be displaced and separated during this process. And this is E-cadherin. And so here this is labeled with GFP, and this is CDH1 GFP in green. And what you can see is that the adhesion molecules are initially fairly homogeneously localized at the cell-cell contact. When the microlumens are appearing, the adhesion molecules are displaced. And this is an overview of the embryo. And what you can see here in this close-up is that initially you have a fairly homogeneous signal of E-cadherin, and eventually when the microlumens are forming, the adhesion molecules are pushed to the sides of the microlumens. And to see whether the fluid is what displaces the adhesion molecule, we blocked the fluid accumulation in 3 different ways, and all of them lead to the same answer, which is that if you don't have fluid accumulation, the adhesion molecules do not move. And this is compatible with the idea that it is the fluid which displaces the adhesion molecule. So if this is indeed hydraulic fracturing, then having less adhesion would in principle allow the fluid to accumulate quicker at the cell-cell contact. And so for this, we used a mutant which lacks the maternal allele of E-cadherin. It still has a paternal allele, so it is viable and develops into a blastocyst and is actually growing into a mouse, which is fertile. And so here on the left side, you have the wild-type embryo. And what you can see from this movie is that the E-cadherin heterozygous mutants, they grow their lumen faster. And the microlumens, they also open up faster. And again, this is compatible with the idea that the fluid is breaking the cell-cell contact. And if you have less adhesion at the cell-cell contact, it will open up faster. And here comes the part where we can now start to ask questions about design principles of morphogenesis. We can now use this tool to engineer the embryo to see if we can actually pattern the mechanical properties of the embryo and control its morphogenesis. So we use those mutants to create chimeric embryos. And so those chimeric embryos are now made out of half wild type and half E-cadherin mutant cells. And so what we expect is that this embryo will have one half, which is less adhesive, and that the fluid, when it comes and accumulates inside the embryo, will break open easier on the side which is mutant, and that in the end, the fluid will accumulate on the mutant side. And so this is a wild-type camera, and so the lumen just appears somewhere, more or less randomly at the trofeculum ICN interface. And then when you engineer the adhesion of the embryo, what you see is that the lumen ends up on the mutant side. And so patterning cell-cell adhesion is sufficient to guide the positioning of the first mammalian lumen and position its first axis of symmetry. So in principle, we could the embryo could control the position of its lumen by controlling cell-cell adhesion. And when we look for any kind of signs of patterning of cell-cell adhesion, for example, if we look at this embryo, it starts to break open a bit faster on the top right. Maybe the lumen will form there, but it forms on the other side, which looked much tighter. And we basically looked for any kind of sign that we could look for in terms of patterning of adhesion, and we could not find anything in relationship to the final position of the blastocell. So we looked for the second phase of the formation of those microlumen, which is when they disappear. And so for this, we started imaging at a higher pace the disappearance of those microlumens. And here is this phenomenon imaged at 5 seconds. And so here you see a fairly large microlumen here, and you see that it grows larger and larger. And then you have smaller microlumens, which are at the cell-cell contact connected to this larger microlumen. And you see that basically they do not coalesce and fuse with this larger microlumen. They just shrink and disappear. And this is reminiscent, this coarsening process is reminiscent of a a process which is called osmotic ripening. So when you have two liquids which phase separate, for example, oil and vinegar, when you make a vinaigrette, you shake it, and eventually the two liquid will phase separate, and you will see droplets of vinegar forming. And the way that the phase separation goes all the way is not by having the vinegar droplet coming into contact and meeting. It is mostly by exchanging fluid at a distance. And this is controlled by the Laplace equation, which relates the surface tension of a droplet to its size and its pressure. And as a result of this, the smaller droplets are under a higher pressure and lose fluid faster than the larger ones. And that's how you accumulate all of the fluid into the largest droplet. And so taking this analogy, we could imagine that we would have several microlumens connected to each other by the intercellular space, and they could exchange fluid based on this Laplace law. And so you can build a simulation in collaboration with, so we did that in collaboration with the lab of Hervé Turlier at the Collège de France. And here you can see that you have two microlumens, and the small one will empty itself into the large one. Because the embryo is not a vinaigrette and can control its surface tension, you can imagine something a bit more different, where you would have a large microlumen with a high surface tension as compared to the smaller one. And then you can actually reverse the fluid flow from the large one into the small one if the cell is controlling specifically the surface tension of a specific microlumens. And so this is also how the embryo could control where the fluid goes eventually. Taking this into account and the connectivity of the microlumens, which is also going to be important, because you can imagine that if you are a central microlumen, you will have many neighboring microlumen to draw water from. And if you're at the periphery, you will have less choice. And so this will affect the dynamics and will affect the final result. So taking a network of microlumens, which are either at the trophectoderm-ICM interface or at the ICM-ICM interface, you can coarsen this in simulation. And if the tension of both tissues is the same, the lumen forms at the ICM-ICM interface, which is not what happens in the embryo. In the embryo, it robustly appears at the trophectoderm-ICM interface. And for this, all what you need to do is to have different surface tensions between the two tissues. The trophectoderm should have a lower surface tension than the ICM. And so effectively what the ICM will do is squish out the fluid away from its cell-cell contact faster than the trophectoderm. And we actually knew that the trophectoderm have a lower surface tension than the ICM cells. This is something that we had measured previously. And you can see that fairly easily by looking at the curvature of the cell-cell contact. Here, if you look at the top left embryo, the ICM cell is bulging into the trophectoderm cells. And this is because they have a higher pressure and a higher surface tension. And this is a result of differences in the contractility of the actomyosin cytoskeleton of those cells. If you block the contractility of the cells using a drug called blebistatin, then the interfaces become flatter. The ICM cells do not bulge any longer into the trophectoderm cells. This curvature of the cell-cell contact, you can also find it back into the microlumens themselves. Microlumens can be fairly symmetric, like this guy, for example, or a bit more asymmetric with a curvature on one side different than from the other. And when they are asymmetric, they tend to bulge into the trophectoderm cell, not the ICM cell, which is telling us again that the ICM cells seem to be under a higher pressure than the trophectoderm cells. And this could, then this pattern could explain how the lumen would form at the trophectoderm ICM interface in a robust manner. To verify this, what we wanted to do again is to engineer the embryo so that we can create a pattern of mechanical properties. This time we use the mutant for MYH9, which is encoding for the non-muscle myosin 2A paralog of the myosin motor. And those guys have a threefold lower surface tension than the wild-type embryos as we have measured. If you create this pattern of contractility, what we expect is that the wild-type cells, which are here in pink, will squish the fluid more effectively than the mutant cells, and this will guide the fluid into the mutant part of the embryo. And this is indeed what we observe. The lumen ends up on the side which is mutant. Patterning contractility, just like patterning adhesion, is sufficient to position the lumen at the trophectoderm ICM interface in this part of the embryo. And so as I was mentioning, so we wanted to understand what position the first lumen in the mammalian embryo, and we think this is mediated by a pattern of contractility with the trophectoderm being less contractile than the ICM cells, and this is ensuring a robust positioning of the lumen at the trophectoderm ICM interface. Otherwise, the homogeneous breaking of cell-cell contact by hydraulic fracturing could takes a risk, put the embryo at a risk that the lumen would form at the ICM interface, which would be bad. So how do you affect this? How do you achieve this robustness? It is in part by having a pattern of contractility. With this, I am finished. I just want to thank the entire lab. And this is a work from Julien Dumortier, who is not on this picture. Francesca has also contributed a lot to the experiments. This is a collaboration with the lab of Hervé Turlier at the Collège de France. And with this, I'm finished. Thank you very much.
Okay, great. Thank you very much, Jean-Léon. Maybe then next, Prisca. I just would like to mention to people that, again, for those that were not here at the beginning, please, if you have any questions, you can start typing them already in the Q&A box at the bottom right, and then we'll sort of ask them afterwards. Okay. All right. Thank you very much, Jean-Léon. Maybe Prisca, you can share.
So it's really nice to be here and to discuss with Jean-Léon and Edward about some of the work we have been working on in the past years on how cell fate coordinate the mechanosmotic forces during organoid crypt formation. What we're mainly interested in the lab, and now it's not working, yes, is to understand how single cell cross biological scales, mainly from individual cells, to create higher order structure that have different functionality and clearly different shapes. And these require a lot of space-time coordination of cells that need to sense, change their shape, and change their function. I'm also very interested in understanding and how these feedback on individual cells, really the feedback between individual cells and their fate and their shape and function. This is why we are really interested in understanding how individual cells transmit information forward. And this is part of also what comes with the Edward's introduction, what are the scales that biological information and where is information encoded that is clearly in chemical gradients, but also in mechanical forces. So you have different waves, both of morphogens, but you also have, and so this is how individual cells generate this emergent property at a higher scale. But then you also need a feedback from the shaped tissue back to the individual cells to coordinate the behaviors. And that is something I think very important to make a system both robust and coordinated. For this, we have been working with the intestinal epithelium, and especially here you see the intestinal epithelium with the stem cell niche that has stem cells and panet cells, transient amplifying cells, and the enterocyte for the full absorption. Already in 2009, Hans Klever showed that a single individual LGR5 is able to form a fully formed organoid. We have an initial step where you have the formation of panet cells, but then you have the formation of what we call the stem cell niche that is able to fold and bend. And this is the only organoid that actually has a shape that recapitulates very much a shape in vivo. Clearly, we miss some in vivo aspects, but this is really a tissue-intrinsic morphogenesis that we can control in vitro. This is how an intestinal organoid grows from an individual cell. This in red is the H2B, and in green you have a plasma membrane marker. You see the cells divide very synchronously. They go from 2 to 4, 8, 16, 32. You have this internal lumen that is created where there's the apical side, and then there's the inflation of this lumen. If you then see there's a contraction of this lumen with the coordinated formation of the same time of the 3 crypts. So you have a very coordinated behavior. So what we have been shown in the past years is that the formation of this organoid is driven, there's no mouse anymore, okay, by a regenerative process that depends on YAP. But then it's clearly that at this point, here, and I cannot show it to you, but really this last point, after the formation of the first panet cell, that is with this notch delta event, you have the formation of the bud. And so this is really where Chotan started in the lab. This is Chotan, she's a postdoc in the lab that is finishing now and is also on the job market. So Chotan really was interested in this step of formation of the crypt. And I really will go a bit into the details. So this is what we have. We have an organoid where we have a stem cell niche. We have a bulging event, that is the second step, where you have this first, in a way, formation of the crypt, but then you have really the crypt formation with the bumping. And again, as I said before, we miss the mesenchyme, we miss a lot of in vivo aspect, but what we are really looking at is with this tissue intrinsic morphogenesis. We are able to see what are the mechanics and chemical cues that are important to form this crypt. This is how it looks like. You have in green the stem cells and beta-catenin in magenta, and you start seeing this first event on the left where you have the first crypt and the niche, then you have the bulging and the budding on the right. Again, you already saw one of these videos, and so what we use for this is this light sheet organoid imaging, and it's very similar to also the microscope Jean-Léon used for his imaging. We have a dual illumination and detection that is really allowing us to have a very high resolution for very long period of time of organoid embedded in matrigel. What we are able then to focus also for the feature extraction is to measure many morphogenesis features like the lumen volume, the tissue volume, the total volume, and the different shapes of the organoid. One clear aspect is that while the crypt is forming, here you see, and now my mouse works, here you see that the cells are triangular. They really start constricting apically. So they start having a triangular shape. And here you can also see it in quantifying on 10,000 organoid, where we can, maybe not 10,000, maybe let's say 1,000, I don't remember. But what you actually see is that here there's this really apical constriction, and the apical domain of the cell in the crypt start being very small. Simultaneously, also quantifying in many different organoids, you see this very strong lumen reduction here in orange, while the total volume stays quite constant. So what's happening, the mechanics during crypt formation is that you have first this apical constriction, and then you have this also lumen volume reduction. This is when we also started collaborating with Edward and Schley, and we started designing a 3D vortex model that really divides the organoid in two parts, the crypt region and the villus region. From all of these, we can input the height of the cells, the apical and the lateral and the basal contraction. And so the three different possible models that Edward and Schley were able to really to design were one where you have differential in plane tension. That means that the full organoid contracts, so then there's like a full in plane tension around. The other one is a differential spontaneous curvature. So this area, the villus region and the crypt region, would then just bend in different ways. The third one would be a boundary tension, so that there would be a line between the crypt and the villus region that would drive the crypt morphogenesis. And clearly, these three models have three properties on the height, also the thickness of the epithelium of the two regions, and the radius. And so this is how the three different models would behave. So to form an organoid with a differential boundary, you would just change in here the thickness ratio, so the thickness of the epithelium, differently than the radiuses. These are all things we can measure in the different organoids. And what was clearly seen is that the only model and the behavior of the thickness of the two regions and the radiuses of the two regions really follow a differential spontaneous curvature model. So for this reason, we also then wanted to measure these aspects, and I'll go back quite fast on the measurement. But so what we were able to measure is on one side, both on the apical side, the cell's tension on the apical side with like laser ablation that is done both on the crypt and on the villus area. And as you can see here, there's like the initial recoil, so that means how fast the tissue opens up when you do the ablation is much higher on the apical side of the crypt. That means that the apical cell, the crypt cells are apically on the tension. On the villus side, on the other way, on the basal side, what we used is something that Jean-Léon developed during his postdoc, and we did it in collaboration with the lab of Takashi. With the pipette aspiration, we were able to measure the basal tension, both of the crypt and on the villus. And in this case, clearly the villus is much more on the tension. When we start looking at the possible molecular players, what was clear is that The myosin here, as you can see in this image, is much higher in the crypt part. So if my mouse still randomly works, yes or no, you see that on the left image on the bottom right, you have the crypt where there's much more apical myosin accumulation. While again, on this image on the top left, you have the villus area where you have much more myosin accumulation on the basal side. So this differential spontaneous curvature is driven by differentially accumulating myosin or on the apical and the basal side. This is also sulfate induced because when we make organoid only from stem cells, the first column, only from Paneth cells, another component of the stem cell niche, you see that myosin here in green is much more accumulated on the apical side, on the inside of the lumen, while in enterocysts that are only enterocyte is on the outside. So what happens is that while the cells differentiate, the stem cell niche start accumulating myosin on the apical side, while on the villus side, there's the accumulation of myosin on the basal, they're giving this spontaneous curvature. But there was still something missing, that is this very strong volume reduction that was coordinating the crypt formation. And so this, we went back to the diagram, to the model, and what the model predicts is that on one side, here you see, really annoyed by this mouse, on the left side, you have on the phase diagram, on the x-axis, you have a normalized volume. On the y-axis, the crypt apical tension. If you see that going up at normalized volume 1, you can go up. So in any case, apical tension is alone able to close the crypt. But the volume reduction really accelerated, because if you then move on the x-axis, you really see that this is maintained. What this model also predicts, that there is a critical value of apical tension at which the organoid is not able to open the crypt again. So this is something, and this is the dashed line. When you go at that level of apical tension, you're not able to open the crypt. And this is also seen in different disease, or also different, for example, infection of the intestine. You can have very strong pressure in the intestine, but the crypt doesn't open up. So it's probably above this value. We were able to then use different experiment model, and I will just show you one. We go above this critical value of apical tension, and then we inflate the organoid. And as you can see here, we can inflate very strongly the lumen without affecting the crypt. That is, now it's work here, bottom right. And here you have it, and this is extremely inflated. So what I showed you at this moment is that the lumen shrinkage accelerates the tissue shape transformation. And there's a critical value of apical tension that really can protect the crypt from the mechanical pressure. Then we wanted to get, and then it's the final part is really to get at the mechanism of this volume reduction, because this means that you have a coordinated behavior of another tissue part, so the villus of the intestine are able to drive a coordinated behavior of the crypt. So we wanted to understand how is this villus behavior affecting the crypt. Again, as I showed you very fast, we can make organoid with stem cells, Paneth cells, or enterocyte. And again, we did all this organoid with a different cell type. And clearly, the one that drives the reduction of the lumen is the enterocyte, so the villus cells. As you can see here, these are organoid made only of enterocyte, and this is a very strong reduction of the lumen. We did some single cell RNA-seq experiment to understand, and here you see it on the left, these are the enterocytes, so this specific cell type that absorbs a lot of the fluid. And we start looking at all different transporters, aquaporins, and see what is specifically expressed in the enterocyte over time. One of these is the sodium glucose transporters, SGLT1. On top of being expressed specifically enterocyte, is specifically expressed on the apical domain of enterocyte. As you can see here in white, they're really expressed on the apical domain here. And when perturbed, they are not able to absorb the liquid and the fluid, and with the change of osmolarity, and there's an inhibition of crypt formation. So what is happening here is that these transporters, this SGLT1, that is a sodium glucose transporter, while transporting sodium and glucose, also imports a lot of water from the lumen into the enterocytes. And this is why we don't see changes on the full volume, because there's a relocalization of the water and of the fluid inside the lumen with the change of osmolarity to the enterocyte. And so to conclude, what the formation of crypt is really driven by both a combination of mechano and osmotic forces. And this is driven by cell fate differentiation. So at a certain point, cells start differentiating to crypt and villus tissue. This initially drives this differential localization of myosin that gives a differential spontaneous curvature. On the other side, as soon the villus start to be formed and the enterocytes start differentiating, we have the expression of membrane transporter, one of which is SGLT1. There might be also others. We didn't look and then characterize all of them, but there might be also other transporter that then drives the lumen shrinkage that accelerate the crypt branching, and not only accelerate, and also coordinates it. This, I think, it's extremely interesting on trying to understand how cell fate can drive crypt branching. But we have also some work, and I'm happy to discuss it, and also probably Edward is happy to discuss, that we know that this crypt branching has a feedback back to the cell fate. And so this is, I think, some of the aspects that we could discuss in understanding how then shape to cell fates, how this branching then creates some robustness in the cell fate and further maturation of different cell fates. And with this, I would like to thank the full lab, but especially Chotan, that is also here, not in the picture, and that also had this very beautiful cover of the paper that was recently published, back to back also with the paper of Xavi Trepat's lab, where they look in 2D at this mechanical, the mechanics of crypt formation on intestinal cells in 2D. And a very interesting news and views of Celeste Nelson that I really advise to read. And with this, very happy to start the discussion with all the others.
Great.
We managed. Okay.
Great. We're actually up to a minute exactly on time. It's amazing. Great.
And I have 25 slides, so I went.
So there's a bunch of questions. I mean, there's a bunch of questions for both speakers. So I'll go. I'll actually go sequentially. So if you want to read them, they're also in the Q&A. So the first one is for Jean-Léon from Martin Lenz, and it's so the also the ripening picture implies that the microlumens essentially exchange fluid with each other. But in this case, there's a big reservoir of fluid outside the embryo. Do you have two reasons to believe that the microlumens are to some extent insulated from the reservoir, which fixes the chemical potential of the water and therefore spoils some aspect of the Ostwald picture? With the second, more specifically, Ostwald is a zero-sum game. If you have a reservoir, you may have a situation where everyone shrinks. Yes, so we have very good reason to know to think that the outside reservoir is isolated, and so these are tight junctions. So it's very important for any lumen that you create those tight compartments, right? And you want to make sure that the osmolarity of both compartments, either on the apical or basal side, the composition should be carefully controlled. And this is the case for many kinds of organs. Think of the kidney, which carefully regulates the chemical content of the blood and of the urine. This is exactly how you do it with tight junctions and selective transport. And so the inside of the embryo is isolated, and then at the same time, what is also there is, and that is regarding your second point, which somewhere went down, which is that there's also active pumping of the osmolyte throughout the process of coarsening of the microlumens. And I think without this, you would not be able to have a proper lumen forming. And I think it's also the case for the organoid of Prisca. I mean, if you don't have tight junctions, then basically it would be difficult to change the. That's actually one question. Maybe for you can have cross comment. It would be nice to cross comment. Actually, maybe Prisca, you can maybe because what happens, right, in the case of the intestinal organoids, is that the fluid is not lost in the outside medium. It goes into
I forgot which one.
Heterocytes.
In the enterocyte cells, right? It's not lost.
And the tight junction there are extremely tight because that's the apical domain of our intestine that is super tight by definition. You don't want to have too many, that is, and clearly you have a very controlled transport, and this is relocalized from the lumen to the cells.
Okay, cool. So actually the next question is for Priska by Florian Schober. Thanks for an inspiring talk. How do you think that the basal lamina and connective tissue in vivo will support or guide apical tension and crypt formation?
In vivo, so clearly there's some work also done with generating some origami that are on below that is really also supporting these aspects. And so part of it is supporting the cell differentiation. So you have some mesenchyme that would support more wind at the crypt that would have more cells and then would drive the apical constriction. In vivo, what is also the difference is that it's an open tube. You don't have a closed tube like the organoid. So I think that would be something to keep in consideration. But probably the changes in epithelium thickness would compensate the volume reduction. So the epithelium thickness of the enterocyte would really, I think, contribute there in vivo for the deformation. But these are extremely very good questions to try to understand also in vivo. And where is the contribution of the mesenchyme? Is it a positive feedback on reinforcing some different patterns that already exist as a tissue intrinsic?
Okay, great. Thank you very much. So the next question is for Jean-Léon by Ninadini Sharma. Beautiful talk. How often can hydraulic fracturing result in cell death by hydraulic instability, etc.? Is it known how cells will try to prevent or control this? If it does exist, is there a benefit to this?
So in the forming blastocyst, there is no need for cell death, and there is no cell death happening at the time of lumen opening. There is a bit of cell death afterward, and that's basically going to be maybe 1 or 2 cells out of 60 plus. So there is very little cell death, and the cell death which is happening is probably not happening because of hydraulic instability. Now, if you're hinting at another point, which is that when the surface cells start to pump osmolytes, this is going to change the direct environment of the cells which are inside the embryo, and this could lead to some osmotic changes, which would maybe change, the behavior of the inner cells. And for example, if you add some osmolyte on top of regular cells, they will start to shrink, right? They will have an osmotic shock, and we do not see much volume change of the inner cells. So I do not think that this is happening.
But can I ask what then drives the death of the cells after?
So this is linked to the differentiation of the inner cell mast cells into either primitive endoderm fate or epiblast fate. So this happens later on. And the primitive endoderm cells, they need to be in contact with the lumen. And so some of those cells which are specified into primitive endoderm within the inner cell mass, but away from the lumen, they will start to migrate or move towards the lumen, and some of them fail, and those which fail, they undergo apoptosis. So this is part of the differentiation process.
Okay. Cool, thank you very much. Next question from Claudio Collinet for Priska. How do enterocytes and stem cells, Paneth cells, sort away from each other in the organoid before crypt formation?
Ciao, Claudio. And it's a super nice question, super good question. I think that this is something we have a full project in the lab really to try to understand. I don't think it's a complete sort mechanism in the sense that they are already formed next to each other, not completely. So the emergence of the two cell type is a bit temporally shift. You first have Paneth cells, then stem cells, but they're always a bit in the neighborhood of each other. But then they create really this coherent stem cell niche. And so this is like how does this stays and how it's stabilized, and it doesn't go all around, because if it's notch delta, you could imagine it goes all around. I think these are very important questions and we still don't know. What we know is that the fate might be reinforced when you create the crypt, that the stem cell in the stem cell niche before forming the crypt might be different from the one later on. There's some work that will be published soon, that of our collaboration we had with Matthias Lutolf, where we look at symmetry breaking, so the formation of this cell fate, and we see they are happening more when the epithelium is bended. So probably what's happening there, that is also feedback, and that is what I was mentioning at the end between the shape and the maturation of the cell type. So I think there's an initial cluster of cells that is an initial niche that is still stabilized by chemical gradients that then is reinforced by the morphogen, the change of the crypt and the robustness. That's work in progress.
Cool. So thanks, great story and super interesting stuff. The next question is Jean-Léon, is for Jean-Léon from Chavet, Trepa. Microlumens form at many different junctions at the same time. What synchronizes their formation across the embryo? So
short answer is we don't know. Then if I can elaborate, basically, I mean, because of the pumping of the fluid and of the osmolyte by the outside cells, we would expect some kind of outside-in wave of breaking of the cell-cell contact, and those microlumens would appear first in the surface cells and a bit inside and then inside. This is not what we see, it just happens everywhere, and it doesn't seem to happen with such a big difference between one side of the embryo versus the other. So we do not know what precisely controls this. Coming back to the first question of Martin, I think in the end, it's about where the tight junctions are. So the tight junctions are the surface, and so this insulates the inside of the embryo from the outside of the embryo. But inside the embryo, we think that the fluid can just freely travel fairly fast between cell-cell contact. So I don't have a clear answer for this, but this is, I think basically the fluid is able to move within the intercellular space very, very fast.
But is there something expressed or something that, you know, at that moment, so if you would look at protein express, you know, protein.
Yeah. So it's not very clear, you know, so, you know, you could imagine that all of the surface cells start to pump at the same time. Mm-hmm. You could imagine just one cell starting to pump, and then it just pumps salt, and the salt diffuses fairly fast throughout the intercellular space. And then this draws water from all cells, right? So this we do not know. We do not know how many surface cells you need to pump. And we do not know what happens if it would only pump in one side, for example. So this is something that we're looking into. Obviously, we've just looked at what we could look at. And, you know, as Edouard was mentioning, you know, the role of hydrostatic and osmotic pressures in shaping organs has had some rebound in the recent years, right? But basically, if you look at morphogenesis paper in the past 20 years, it's all about actomizing and adhesion. And so we looked at this because the tools are there. But we need to develop new tools to engineer the pumping ability of the cells. I think that would be very important for studying those kind of processes.
And also fluorescent reporter, where you can measure almost osmolarity. That would be, you know, some things like we can measure in a way still pH quite decently. I think for others it's not that trivial. And
I'm calling out now anyone who is good at optogenetics to engineer any kind of ion channel that we can open and close with light that would allow us to pump fluid. I mean, that would be just awesome.
Yes, I just follow.
Firstly, in terms of synchronization of lumen, I mean, I guess villus cells alone do it. So you would also, I mean, you would also think it's cell autonomous based on fate, or that there's a synchronization or active, or what do you think?
Yes, so I think it's cell autonomous for the expression, but again, I think the differentiation of enterocyte is not fully cell autonomous. We randomly see like one that just do it. It's normally then at a certain point, some cells start both to express some genes, start moving in a different way. So there's just a differentiation to enterocyte seems already to be collectively, but per se the expression is cell intrinsic, but that gives a phenomenon that is collective, because collectively the change in epithelium happens all over at the same time. We didn't try to have like mosaic with like few cells that will express it, and if these would be enough to then drive it. I think that would be quite interesting also to see which other. So we tried the aquaporins, and that didn't give a very strong phenotype. They're not also super highly specifically expressed on the optical domain, but there might be also other transporters that affect this. And the other aspect that I was really mentioning going back is this coordination of crypt formation. So it's known in vivo that crypt are formed very synchronously in the full intestine. So how do you make this very synchronous behavior? So I think that and it's coming very, it correlates with the appearance of mature enterocytes that also compacts. And so we have been seeing this in vivo, that compaction and increased epithelial thickness in vivo is like correlates very well with the appearance of crypt. But to really go to the causality, it would be, I think, really interesting.
Cool, thanks a lot. There's two questions. So there's two questions left. One, one for the next one for Priska by Matreyi Narasimha. Does the membrane transporter in the villus influence actomyosin contractility in the crypt?
Yes, we are looking into it in the sense that we are looking at the transporter, not directly the transporter, but the changes in lumen affect the actomyosin. So we are redoing the experiment of the inflation and the reduction of the lumen and looking at the myosin relocalization. And there's clearly a feedback with the volume and the localization. But maybe this even Edward can say more if he wants.
Yeah, we're quite interested.
So on the relocalization of the myosin, so I think there it would be something interesting to look at the feedback, especially on fate and the relocalization of myosin.
Yeah. And I guess for us, we were discussing about this with Priska a lot, because then it's very tempting to think that there's mechanotransduction in there, but then of course what is sensed is its cell shape, is it stress, is it curvature? I mean, that's the hard thing, I guess, but that's exactly
to see exactly what is happening. So we're modeling and
And actually, if someone is interested, I mean, Elias Barga also commented on this, a reference that looks that I didn't know, but that looks interesting. So on bioarchive, so thanks.
Thank you, Elias.
Thanks, Elias. Okay, okay, Jean-Léon is gone.
Xavi's comment that Xavi soon the room will be open also to people to become open. So maybe you can join us. But I think the general comment of Xavi, we can wait also that Jean-Léon is back.
Maybe that makes sense actually. There is a question in chat actually, maybe that's a bit more specific that we can address now before going to the full discussion, from Alessandra Agresti, wondering how all of these osmotic tensions affect the nucleus position, shape, DNA replication, transcription, and so on. I don't know if Jean-Léon or Priska want to.
So I was disconnected for a minute or so. I didn't hear. I'm sorry.
No, but so if there's changes, how the osmotic forces and the different change in tension affect nucleus. There were two very nice papers also published, how nuclear sense the external environment, published last year, not from our labs. And I think that would be, but there is a lot there to work on. And then there's clearly change in shape and the correlation. And there are, I think, data that shows that there's clearly correlation between nuclear shape and cell shape, but it's not clearly if it's one-to-one. And with some experiment that I at least did in the past, I think the correlation works well after division, but then if this changes over time, I think would be interesting to look at what's the correlation with these forces and the transcription.
Cool. And then indeed,
there was a question, and Edward from Xavi.
Well, I mean, then there's general. I mean, so there's two general questions from Xavi Trepa and David Sprinzak that maybe actually at some point we can open up to everyone. So the first one is, how general is hydraulic fracturing as a mechanism for luminogenesis? And the second one is sort of what's your take on the role of cell adhesion forces versus actomyosin tension in regulating morphogenesis? Are there rules for where there is one over the other? Tough questions.
Yeah.
On the first one, I can say we have been looking at the formation of lumen in organoids. And this is
tough questions.
Yeah. On the first one, I can say we have been looking at the formation of lumen in organoids. And this is probably happening almost at the single cell stage, single cells with a lumen. So there, at least some hypothesis, but it might be more trafficking and polarized trafficking of vesicle, that because the organoid is embedded in matrix, it's all basolateral and these apical vesicles maybe fuse. So might be a bit more trafficking, but we never also looked at very high spatial temporal imaging to see if there's something also, something else on the memory. So at least on the lumen of the organoid seems not to be, but that's the organoid.
And Jolyon, yeah, adhesion versus tension. Tension. You're probably the what's the best?
I mean, it depends what you mean, right, by adhesion. But I think that was nicely hinted by Edouard at the beginning. Is animal cells have at their disposal several different tools to drive morphogenetic movements. And contractility is one of them. Adhesion is one of them. Turns out, changes in osmolarity and changes in hydrostatic pressure is also another tool that the animal cells can use at their advantage in different settings to shape different organs. And this is all about now, the power of the in vitro studies and organoids and so on and so forth is that you can challenge the cells and the tissues and put them into settings which will, cause them to do maybe extreme versions of things that they would normally do during normal development or during pathologies and so on and so forth. And then basically what we do in the lab is we try to understand between those different tools that the cells have at their disposal, which one do they use to undergo this specific morphogenetic movement. But whether actomycin is more prominent than adhesion or whatever, I think it just all depends also on the tools that we have to study them, because we have been looking at a lot at actomycin and at adhesion because we have the tools for it. That's it.
I mean, thank you very much. So that's great, because I think we're at 3. I mean, we're at 3 p.m. and we're basically finished all the questions. So I think that's perfect. So yeah, I just want to thank again the speakers. I mean, both for great talks. And now, if I remember the program correctly, we'll move towards sort of more, let's say, smaller committee where everyone is invited on stage, and we'll try to manage a global discussion